Hello students, welcome back. Welcome to today's NEET Biology live class. So you remember yesterday we were discussing about immune system, right? So in that, I think I have shown you this picture. Remember this? So students who have attended yesterday's class, okay. So you remember this picture. What was I talking about yesterday? I said that if at all a pathogen or a microorganism comes from outside as a foreign body into our body, then immediately there is a set of class of immune cells which can recognize them as foreign bodies, right? And they can actually get hold of this foreign body, and they can get it and present it to another set of cells. Those another set of cells we will see later, but right now the initial cells which come in contact with the foreign antigen, these cells are called as antigen presenting cells, APC. Understood? So which cells can behave as APCs? Do you remember? Let me see if you remember. I'm waiting for this answer. Because yesterday one student was saying, I think Varalakshmi, that I am not asking questions. Yesterday there was lot of content. You understand, right? There was lot of syllabus and I was more into explaining those intricate concepts to you. So we had little less questions, but don't worry, today I'll be asking you questions also. So here is my first question. First of all, you tell me which cells can act as APCs. This is an important question. Which cells in our body can act like antigen presenting cells? Waiting for your answers. Yeah, there is an answer, lymphocyte. Himanshu says lymphocyte. So lymphocyte is a very broad, general or a vague answer. Specifically one kind of lymphocyte. Which one is it? Someone said WBC. Well, white blood cells are leukocytes. No. So I'll tell you again. Maybe you people have not attended my class yesterday, maybe. All cells other than RBC. Who is this? Sai. Sai has answered all cells other than RBC. You're right, but you're talking about ASCs, which are, I'll come to that. Okay, even you are right in one way. But right now I'm talking about antigen presenting cells. These are special immune cells, right? If you're saying all cells other than RBC, then you're talking about all cells of your body. All cells of your body are not immune, right? They are not immune cells. So they are normal cells. Of course, even normal cells can present antigen, but how they present, what kind of antigen they present, I'll tell you. Right now I'm talking about the antigen presenting cells. These cells can recognize exogenous antigens. Exogenous means outside. Those antigens that come from outside, as soon as they enter your body, these cells can go and get hold of them and present it to higher cells. So these are APCs. Normal cells cannot do this, right? Normal cells can only present endogenous antigens. That means if they are virally infected, if the virus goes inside the cell and it starts multiplying, then that viral particle will be presented by the cell. That is endogenous antigen. But exogenous antigen can be presented only by antigen presenting cells. These cells are B lymphocytes. B lymphocytes and dendritic cells and macrophages. Well, these three cells, their original function is not APC, not antigen presentation. Antigen presentation is one of the accessory functions. Their main functions you already know. B lymphocytes, they produce plasma cells, and plasma cells produce antibodies. So that is their main function, right? Same way macrophages or dendritic cells, even macrophages, you see phagocytosis. So these are the main functions and these are the accessory functions. Okay, so well, now whenever an antigen comes, the antigen presenting cell will take that antigen and it will present on this MHC2 molecule. MHC2 molecule, where is it present? MHC2 molecule is present on the surface of the APC only, right? Normal cells have MHC1, but APC is also one of your body cell. So when just now Sai answered saying all the cells of our body except RBC will present antigen. Well, why did he answer this? Because yesterday I told you all cells of your body have a glycoprotein on their surface called MHC1. Understand? All cells. Whereas special immune cells called antigen presenting cells, they also have MHC1 because they are also cells of your body. Along with MHC1, they have an extra glycoprotein molecule called MHC2. Okay, it is like if you join the police department, you will have an extra uniform, police uniform. Normal people have their own dresses, right? The clothes that we wear. So even the police have the normal clothes that they wear. Along with that, they have an extra uniform. That's the police uniform. Same way, all cells, APCs also have MHC1, just like any other cell. But extra protein, glycoprotein molecule that they have is MHC2 because they are antigen presenting cells. Okay, and this MHC2 is specialized in capturing an exogenous antigen that comes from outside the body, and that is why it is called as antigen presenting cell. So clear with this. Now, what happens and what does it do when it takes this antigen? After taking the antigen, where is it going? Let us see. So now you can see this. APC has taken the antigen and it is moving to the police station. Then it goes here and it attaches to something. Can you see that? Which cell is this? T helper cell. T lymphocytes, you know the types, right? T helper cell. CTL means cytotoxic T cell, right? So T lymphocytes, cytotoxic T lymphocytes, or TC cell we generally write. TC cell we call it, no? It is the same cell, TC cell. Okay. Fine. So what happened here? Please observe this. This APC with MHC2 and an antigen bound there, it has gone and got connected to T helper cell. T helper cell had a receptor on it. Can you notice the receptor? I'll remove this first. Let us study T helper cell. T helper cell has this molecule, right? This molecule. Can you see this? A molecule attached on it, surface. That molecule is called as CD4. CD4, okay? And this is another molecule. This is basically a receptor. It is called as TCR, means T cell receptor. Okay, T cell receptor, because this is a T cell, T helper cell. T helper cell has what? T helper cell has TCR and CD4. Wait, now you see, if you classify T lymphocytes broadly into two types. Of course, other types we are not really talking about right now. We are only talking about helper cell and cytotoxic T cells. You must have read this in your book also, right? So what is the difference between them is helper cells on their surface have CD4 molecules. CD means cluster differentiation, okay? Cluster differentiation factor 4. That's the name given to it. It looks different from CD8. Whereas cytotoxic cells, you see their structure, they have a CD8 molecule. CD4 is different from CD8. That is how they both are different. Of course, their functions are also different. You know what is the function of cytotoxic T cell? Cytotoxic, it is toxic to cells, means it can kill the foreign antigen cells, right? So we'll deal with that later. Right now you understood the difference between these two T cells. One is helper, one is cytotoxic. Both have, one has CD4, another has CD8. Helper has CD4, cytotoxic has CD8. Remember this. Okay. Now, when this goes like this, I think we need to draw an antigen in this, right? Yes. So with the antigen, when it goes, that antigen, it is presenting now, because this is an APC, antigen presenting cell, it is presenting that antigen to the T helper cell, and T helper cell will receive that antigen using its TCR, means T cell receptor. So who is binding to whom? Carefully observe. The MHC2 is binding to both CD4 molecule as well as TCR. It is binding to TCR through the medium of antigen, keeping the antigen in between. This is MHC carrying the antigen. From there TCR binds like this. So MHC2 with the antigen is presented to TCR, that is T cell receptor. Whereas at the same time CD4 comes and binds to MHC2. Now why two things are binding to MHC2? The thing is this. CD4 will first detect. First who is binding? You can see. See, even before, let us say you are here. Even before MHC2 went and it got bound to TCR, CD4 is first of all binding it. First it is binding, right? After that MHC2 is binding to TCR. So CD4 can recognize MHC2. What is the purpose of CD4? CD4 will recognize MHC2. Okay. CD4 cannot recognize MHC1. Understand? If MHC1 came and bound with its antigen, CD4 cannot recognize. So T helper cell will say, no, this case is not, it does not come in our area. It is not, you have to go to another police station. Okay, that is what it says. So it says we recognize or we take cases that come only through MHC2. Who will check that if the case is coming through MHC2 or MHC1? CD4. First CD4 will bind with MHC2 and it will detect. Then MHC2 will present the antigen to TCR. Then T helper cell gets activated. Okay. And T helper cell gets multiplied. So proliferation of T helper cell is what we say. So you get many T helper cells. Not only that, these T helper cells will now produce substances called cytokines. What are these called? Cytokines or lymphokines. Cytokines because they act as messengers or mediators between cells. Okay. So cytokines. These cytokines will go and they stimulate either B cell or TC cell. This is TC cell. So now the question is, T helper cell will take the case, right? It is taking the case, antigen, and it is thinking, oh, this antigen, is it small or big? Is it, what kind of is it? It will process, T helper cell will process the antigen, then it will decide, shall I send this case or this antigen to B cell or shall I send it to TC cell? Who will take the call? T helper cell. That is why it's called helper cell. It is helping us to decide where the antigen has to be sent, what kind of punishment we have to give to the antigen. If the antigen, if B cell is activated, let us say, the cytokine, let us say, goes and activates the B cell, then B cell will proliferate, it will produce plasma cells. First of all, whenever the T helper cells are stimulated, they first produce memory cells, so that next time when the same antigen comes, they will be readily reacting. Memory cells. This always happens. Wherever immune cell is activated, it produces a memory cell first to remember that, oh, this antigen was brought to me. Later in future, if the same antigen comes, these cells are quickly responding. Okay, that is the purpose. That is why we say this specific immunity has memory, right? Non-specific immunity doesn't have memory. So this is very important. Okay, now what happens? B cell produces plasma cells and they produce antibodies. What will antibodies do? They are Y-shaped. Y-shaped antibodies are there, right? They go and they bind with the antigen and neutralize it. Okay, antigen antibody reaction or complex. So if it is small enough, maybe antibodies are produced, they will go and bind to the antigen. Or if this thinks that no, B cell cannot, antibodies cannot kill our antigen needs a stronger treatment, then helper cell will send cytokines to TC cell. Or what is TC cell? Cytotoxic T lymphocyte. What will this do? What is its mechanism? You see, I have even drawn a cytokine receptor here. This one. So the cytokine can come and bind here. Right. Now, what is the mechanism by which this can kill the foreign antigens? It has two mechanisms. One is by producing perforins, enzymes called perforins or granzymes. One is perforins, another is granzymes. Who produces perforins and granzymes? This TC cell. What are these? Why is it called perforin? This perforin will go, for example, if this antigen was a bacterium, then the perforin released by the TC cell, it will go and perforate, means it will make holes in the cell wall of the bacteria. Okay, it will make holes in the cell of the cell wall and cell membrane of the bacterium. Because of that, what happens? Water and other substances, other fluids will enter the bacterial cell and it will burst. So thereby bacterium is killed. This is one mechanism. Or it produces granzymes. Granzymes are hydrolytic enzymes. Granzymes are enzymes which go and entirely digest the foreign microorganism. So very powerful. Which one? TC cell. TC cell's punishment is very stringent and powerful. Whereas B lymphocyte can only produce antibody. That antibody goes and binds with the antigen, right? And antibody production takes a little time also. So I hope you have understood this mechanism. Now, we studied about what happens if an antigen comes from outside, means exogenous. Now we study about what happens when an antigen is endogenous. Endogenous means, again I told you, let us say this is, first let me erase all these things. So this is our normal cell here. Normal cell of the body, not a police cell. This is a police cell, I told you, APC is antigen presenting cell. Now what happens if a normal cell is infected with virus, let us say. That virus went inside and it started getting multiplied. It is multiplying in numbers. Then one of the peptides, protein coat will be there for the virus. That peptide molecule is pulled out of the cell and it will be presented on this. On what? MHC1. Because I told you all cells of your body, normal cells, have MHC1. That means even you have a facility. See, it's just like that. If a criminal comes, policeman can catch the criminal and present him to the police station, right? But even you, as a normal civilian, if you see a criminal, even you can catch the criminal and give him to the police station, right? That is what we are discussing here. Even normal cells also have this function. But a policeman will do it with his uniform. That means he will have the handcuffs like MHC2, special material. He'll take him in the police jeep. But you might not do that. You might do it in a different way. You have MHC1. Okay, you have to present this antigen on the MHC1 and take it to the higher level. Here the difference is a policeman or an APC can take the antigen, present it to T helper cell, but we don't present. Normal cells don't present to the T helper cell. Normal cells directly present to the TC cells. It is like, if it is a department fellow, that guy will first approach his higher officer and take his help. He'll tell him, I got an antigen, help me out. Where do you think I should send this? Do you think it requires B cell or do you think it requires TC cell? You take the call. Because my job is just to catch and give you, because he has to give you and again go back and find other criminals. But we, as normal cells are like, I don't require T helper cell. I will directly want the highest punishment to be given. Emotional people, you see, generally normal people, what they see, if they see a criminal, they say, you have to hang him, you have to kill him. So that kind of attitude. The normal cells, they are directly taking the antigen to the most dangerous fellow, that is TC cell, right? Directly to the TC cell they are taking the antigen. And now we'll see how it happens. So a normal cell is taking the antigen and it is going and it is attaching here, right? So you can see what's happening here. Now you see, when it attaches like this, let me get these things a little bit higher. Just like what happened here in the helper case, you see first CD8 molecule is coming and binding with MHC1, understand? So MHC1, understand? First CD8 will detect the MHC1. Remember, CD4 can detect MHC2, CD8 will detect MHC1. So CD8 will detect, it will bind and say yes, MHC1 has come, you may react. Now the TCR, the T cell receptor will bind this antigen, it will detect it. And it will do the necessary, whatever is required. What it will do? It will produce perforins, granzymes, and those will directly come and attack this own cell, because this is an infected cell with virus. That cell cannot be cleaned off the virus, it has to be killed. That is the only way it has to function. So these perforins, granzymes will come and kill this cell, so that once this cell is cleared, other cells will not be infected by virus. So this is the agenda. Okay, you understand how this happens? MHC1 is going to CD8, that is TC cell directly. Now what is this cell called as? We called it a normal cell. Well, it is a normal cell until it is infected. Once it is infected, its condition inside is altered, right? You can see inside the cell now you have viral particles. So altered cell is called as altered self cell. Altered means changed. Self cell means our self cell, not foreign body cell, our cell, self cell. ASC. You understand why it is called ASC now? This was called APC because it was antigen presenting cell. This is called ASC because it is altered self cell. Every altered self cell has this possibility and chance to present its own antigen to TC cell. So this system, this police system is very fair, just like how our police system is in the society, right? If you want, you can go file a case on somebody, or you may go through the police station. Yes, you can directly give it to the police station. Remember one thing, even APC also, if the APC itself is infected, APC also can present an antigen on MHC1 instead of MHC2. Then that APC will come and present the antigen to whom? TC cell. Because in this case I told you the antigen is being presented through MHC1. So whoever it is comes with an antigen on MHC1 molecule, they have to present it only to the cytotoxic T cell. That is the concept. Okay. Yeah, so someone, Manorama, I think, has asked, how can I watch the yesterday's class? You can watch it in our videos in the Toppr. If you are using the app, you can watch, you can scroll down, because today's videos are on the top. Below that you have all the previous videos. But on the app, if the videos are not visible, you can get them on YouTube itself. In the YouTube, you can click on the channel icon. You have, Toppr icon in below the video. Below my video, you can see the Toppr icon. You can click on that. If you click on that, then you have the video section. Click on the video section. You'll have all our videos. Every video that was streamed yesterday, day before, and all. Not only biology, you have physics, chemistry also. Okay. So please go through them. If you are regularly coming to the classes, you'll have more benefit. So I'm just seeing if I have to tell you something extra here or anything is missing. Fine, I think this must do. This is much more than what your book has given. I just told you so that you'll get some better knowledge about how the immune system functions. And the next topic I want to start is cancer biology. We'll deal with cancer biology. Okay, what is a cancer? So as you know, cancer is one of the leading killer diseases in the world. It's killing so many people, okay, across the world, and still there is no proper cure for cancer. Although various modalities of treatment have come, still only if the cancer is detected in the early stages, the person can be saved. Otherwise, the life expectancy of the person with cancer, usually I'm talking about the malignant cancers, not the benign ones, because benign cancers are not harmful, malignant ones are the harmful. So the life expectancy is low if the cancer is detected at a later stage. So science is working on this to find a good proper cure for cancer. So let us first try to understand, if you want to, maybe you'll become a doctor, and in future maybe you'll also contribute something towards cancer treatment. So for that, first we need to understand how cancer occurs, right? So for example, benign cancer examples are uterine fibroids, and pre-malignant and malignant are two conditions. What is pre-malignant condition? First of all, malignant cancer is that cancer which is the harmful cancer that we say. Benign is something which is not harmful, because this benign one does not spread anywhere. Malignant one spreads everywhere. When it spreads everywhere, that is where it creates the problems. Okay. Benign ones usually don't grow so much like the malignant ones. They grow to some extent and they may stop their growth there, right? And they are localized, means they are at one place. Whereas malignant ones, they keep growing continuously and they get divided and they get spread into all parts of the body. So that is the problem with malignant cancers. Generally, a term called neoplasm is used in cancer. What is this neoplasm? New cells. Okay, new cells that are formed in cancer. That growth of cancer tissue is called neoplasm. That growth becomes like a swelling in the body wherever it grows. See, any part of your body, whether it is inside the bone marrow or on the skin, anywhere, if new cells are forming, obviously those cells will form a swelling, isn't it? That swelling is called tumor. Yesterday class I told you swelling is called tumor, right? So tumor is what is formed in the body. Okay, tumor is one of the essential features of cancer. Hence, sometimes some people use tumor for cancer. Okay. But there are some diseases like leukemia. Okay, leukemia, the blood cancers, usually you don't see any swelling at one place. So generally we can say they don't form tumor. Okay. So what is a branch of medicine that deals with cancers? Oncology. That includes study of cancers and their treatment, everything. So basically we are thinking, even though the mechanism is not clearly understood, but science agrees to this point that cancers are actually caused by failure of cell cycle regulation. When the cell cycle is happening, if that is not properly regulated, cells go on dividing continuously without any stop. So when they go on dividing continuously, then extra tissue is formed, which is what we are calling as cancer. So ultimately it is about inability or a failure of cell cycle regulation. Now we talk about certain things. The first thing we are talking about is contact inhibition. So the first thing I am talking about is contact inhibition. What is this phenomenon? See, what happens is, let us say you take a bowl, and in that you put some cells. These cells are allowed to multiply. Okay, when they multiply, okay, and when a cell multiplies and it produces other cells, all those cells will come and lie around this cell, right? New cells that are formed will be here. Like this, when a cell is touched by all these cells, new cells that are formed from it, Okay, when the cell comes in contact with other cells around it from all sides, then that means this cell has divided, and it has produced more cells. All the other new cells which are formed, they are surrounding this old cell now, right? Now this cell is coming in contact with all these cells. Because of coming in contact with them, now this cell will stop dividing. You understand this? Maybe this mechanism is a natural and normal process. What is happening? A cell, when it divides enough such that the newly formed cells come in contact with this particular cell on all sides, that is a signal for this cell to stop dividing. Why? Because it has understood that, okay, now I am surrounded by cells on all sides. I can touch everywhere, I can touch cells. That means I have produced enough number of cells. Let me stop dividing. This sense of stopping or inhibiting your cell division when you come in contact with all cells on all sides is called contact inhibition, right? That means cell division inhibition when you come in contact with other cells. What is it called? Contact inhibition. Is it normal? Yes, it is normal. Contact inhibition is a normal mechanism, normal phenomenon of cells, and it is very much needed. If the cell does not realize that it has already divided and produced enough number of cells, it will go on dividing. Who will stop it? Nothing will stop it. It will go on dividing. That is what gives rise to cancer, right? So now you tell me, contact inhibition is good or not? Is it good or not? Someone has asked about PCOS, polycystic ovarian syndrome. It's not a cancer, it's a benign condition only. There are cyst, ovaries develop cyst-like condition, okay, cyst formation. So it's not cancer, it's a benign one. It's not malignant. Well, so my question is contact inhibition good or not? Very good. So you have said it is necessary and it is good. It is good. Now, when cancer happens, it happens because of failure of contact inhibition. So remember this. Sometimes question might come like this: which of the following causes cancer? Contact inhibition, etc., X, Y, Z, whatever. You might quickly answer contact inhibition. No. Contact inhibition does not cause cancer. Cancer comes when the contact inhibition fails. So remember this. So contact inhibition's failure causes unlimited growth and neoplasia, right? Now, there is another mechanism that we need to know. Another mechanism is anchorage dependence. What is this? Anchorage dependence. If you take a medium, okay, if you take a bowl, and in this bowl, let us say, you have put some cells. These cells are dividing. They are multiplying and producing more number of cells. So what's happening here is, if these are normal cells of your body, automatically this bottom cells will get sticky and they will attach to the bottom of the bowl that you have taken. Okay, the vessel which you have taken, to that vessel automatically this bottom cells will attach, right? So why do they attach? Because the whole cell mass, if it does not attach, the whole cell mass can move and go to some other place. So for the whole cell mass to be anchored to the ground, it is getting sticky. The bottom layer is getting sticky and it is attaching. Therefore, this term is called as anchorage dependence. What is this phenomenon called? Anchorage dependence. So the entire cells are dependent on the bottom layer, which is anchoring to the ground, right? Which is anchoring to the bottom of the vessel that we are using, the culture medium. This is anchorage dependence. Not only this, this is one way by which the entire cell mass is attaching to the vessel Another thing is, between the individual cells also, there are connections. There are molecules called cadherin molecules. Cadherin molecules which bind one cell with another. One cell with its neighbor cell are attached through these cadherin molecules. Okay. This way, what is achieved now? The entire cell mass, what we have done is, it's like, let us say there are apples. Okay, you have put in a vessel. All the bottom layer of apples you have fixed. Okay. And over these bottom layer, you have put more apples. And between every two apples, or with one apple with the other apples, you have tied small threads. So all apples are connected. Now this entire apple mass is lying on the bottom layer of apples. Now this entire mass will become one single mass, right? Because if you try to move it also, the bottom layer is fixed, so apples don't move. And with the bottom layer, the layer above it is connected through small threads. That layer is connected to another. So every apple is connected to all apples around it. Because of this, what's happening? The entire mass of apples is not able to move independently. They will stay at one place. This is a normal mechanism. Same way, in your tissues, in your body, cells are not only attached to one another, the bottom layer of cells is also attached to the medium, means the surface. Surface, it may be the organ surface, body wall, wherever it is. Okay. So these are two things you have to remember. One is anchorage dependence, which is the bottom layer sticking to the surface. Another is cadherin molecules, where individual cells are sticking to one another. These two phenomena, if they fail, what happens? Let us say anchorage dependence is not there. Entire cell mass can be detached from that point, and it may move to another place. What do we call it? Metastasis. Metastasis. Right. Or let us say anchorage dependence is okay, but these individual cells are not attached to one another through cadherin molecules. Then also, some cells may get detached and they may move to some other place in your body, some other organ, some other region. That is also metastasis, right? Metastasis does not mean the entire tissue has to move. Some small cell group of cells also can move. So the question is, the failure of which mechanisms makes cancer the metastatic, or makes metastasis possible? What is your answer? Failure of which mechanisms makes metastasis possible? Loss of anchorage dependence or anchorage dependence? Loss of anchorage dependence and failure of cadherin molecules, lack of cadherin molecules. Remember, it is a loss of anchorage dependence, loss of contact inhibition, loss of cadherin molecules. Okay, it is their loss. If they are present, it will be normal. So please remember this. Now what is the difference now? Now again I'll ask you a question. What happens if contact inhibition fails? What happens if contact inhibition fails? Neoplasia, metastasis, both, none. Give me the answer. What happens if contact inhibition fails? Neoplasia, metastasis, both, none. It is not both. Some students are answering both. It is neoplasia. Whoever has answered neoplasia, it is right. So Meena and Uma Maheshwari and Spurti. So neoplasia, they are answering. That's right. Because I told you, contact inhibition, when that contact inhibition is lost, what happens? When the contact inhibition is lost, cells don't stop division, they continue to divide, which is neoplasia. It has got nothing to do with metastasis. Okay, metastasis is when the anchorage dependence fails and the cadherins are lost. So please try to understand these concepts and apply them properly when the questions are asked. Okay. Okay. Now, apart from that, we have to understand about a few other things. What are proto-oncogenes? What are oncogenes? And what is p53 gene? In our DNA, we have something called p53 gene. It is a gene. This gene, when it transcribes into mRNA and translates into a protein, you get a protein called as p53 protein. p53 protein. What is the function of this p53 protein? It inhibits the development and growth of tumors. Okay. So p53 is called as guardian angel of our genome. Understand? It is called as the guardian. It means it is protecting, like an angel. Protecting what? The DNA, the genome, the genetic material. Why? Let us see why. Because p53 is the one which is also called as tumor suppressor gene. There are many other genes like this. One example is given in our book. It is called p53. The p53 is a tumor suppressor gene. Means it will not let the tumor to form. It suppresses the formation of tumor. How does it do this? First of all, oncogenes means the genes in your body, in your genetic material, which are cancerous. Means these genes are causing unlimited cell division, right? Oncogenes. Oncogenes are not natural in your body. They are not normal. We don't have normally oncogenes, but we have some genes called proto-oncogenes. What are proto-oncogenes? These are some genes in our genetic material, in our DNA, which have the potential to become oncogenes. Means they are vulnerable genes. For example, see, there might be a gene. If I cause a mutation in that gene, okay, that gene's base sequence changes, right? Still, that gene might not become a harmful one, okay. Whereas some genes in our body, they might be very sensitive, vulnerable. Like even if one base pair changes, maybe it will become harmful. It can produce, it can go and cause unlimited cell division and cause neoplasia. So such genes where more vulnerability is there for mutation, those genes are called as proto-oncogenes. So in case these genes come across or exposed to any factors that cause cancer, like physical factors. What are the physical factors? Like sunlight, the ultraviolet radiation, okay, excessive heat, so many, smoking, it's also a chemical factor. So chemical factors also, if you see, uh, various kinds of chemicals that can cause cancers. So physical, chemical and other factors, whenever to these factors these genes are exposed, what happens is these genes quickly turn into oncogenes. So proto-oncogenes can turn into oncogenes, understand? And the thing is that mutations of proto-oncogenes are dominant. What does it mean? A single copy of the oncogene also can lead to cancer, right? You know, in DNA, the chromosomes, we have homologous chromosomes. So even if one chromosome is exposed to the cancer causing factor, in that if the allele, one allele also changes, and the mutation is occurs in that proto-oncogene, then that mutation, whatever, even if it changes in one chromosome, still it plays a dominant role, and the person can still get cancer. So that is the effect. But still, we have a guardian angel which tries to prevent this thing from happening, from proto-oncogenes to turn oncogenes. Who is that guardian angel? It is P53, right? P53 protein, which is prepared or which is coded by the P53 gene. Now, somebody might ask a question: what if a mutation occurs in the P53 gene itself? You are telling us that P53 gene will protect the entire genome by suppressing the tumor formation. You might ask me, what if somebody causes mutation in the P53 itself? Luckily enough, in the case of tumor suppressor genes like P53, both copies of the gene in the homologous pair of chromosomes must undergo mutation for the cancer to develop. So that is very fortunate that in the homologous pair of chromosomes of where P53 is present, both alleles must get mutated. You have to mutate both the alleles. Only then P53 will fail and cancer will develop. But whereas in proto-oncogenes, which become oncogenes, even if one changes into undergoes mutation, even then cancer can develop. But the one who is protecting us, the P53, for that, if the protector has to fail, you have to mutate both of them. So this is a little assuring for us, right? Okay, now let us see where this P53 functions. This P53's role is something you can see in the G1 checkpoint of the cell cycle. So the G1 phase, S phase, G2 phase, remember? In the G1 phase I told you there is a checkpoint. Again I told you there is another checkpoint in G2 phase. Remember my cell division class if you attended it. And in the G1 checkpoint, at that checkpoint when the cell is going through that, the DNA is going through that, the P53 sits there, okay, and it asks the DNA, come here, I have to check you. Then it checks the DNA. If it finds that the DNA has some kind of damage, some mutation, some damage, you know what it does? You are disqualified. I won't allow you to enter the S phase. Why? Because once G1 phase is passed and you enter the S phase, in the S phase what happens? The DNA gets doubled. Then you enter G2 phase, then mitotic phase, then you go divide. So if you divide, let us say in this cell DNA is already damaged. If you let this cell to pass the S phase and enter mitosis, it will produce two cells which have damaged DNA. That damaged DNA will go on multiplying, right? That should not happen. So our P53 is sitting, okay, it is sitting in between. It is sitting at the G1 checkpoint and it is checking every DNA that is going from there. It is asking, come here, I have to check you, just like our traffic policeman. Okay, it stops there, it has a checkpoint there, okay, and it checks every DNA. Okay, your code is perfect, you may go. No, your code has some damage. So what will it do? Let us imagine some cell has a DNA which is damaged. What do you think the P53 gene will do? You know what it does? It tells your DNA is damaged here. I cannot allow you to enter S phase. Because if you go into S phase, you'll double your DNA, then you'll enter the mitosis and you'll become large numbers, which I don't want to happen. So I have the only solution for you is you have to die. That is what it does. So P53 induces apoptosis in the cell that has damaged DNA. So automatically that DNA and that cell are eliminated so that they don't remain in your body. So damaged cells, damaged DNA cells are not remaining. That is how P53 is protecting us, right? Hence it is called as tumor suppressor gene and guardian angel of the cell genome. Okay, so these are the points I wanted to talk about in the cancer topic. Now we are going to talk about genetics. So well, genetics, a lot to be discussed in genetics. It's a very huge, vast chapter, I understand, but the things that are generally tough or difficult for the students, those things I'm going to deal with them here. Okay. So first of all, let us try to understand a little bit about the terminology. I hope you know the meanings of all these terms. Even I covered these in the cell division, so I'll not take much time for it because I covered them in detail so that it will help you for the genetics part. And there are other things like independent assortment, recombination, linkage, epistasis, hypostasis, so many things which I'm not going to talk about here. First, let us understand the basics. Okay, chromosome. What is a chromosome? If you take any cell of your body, let us say this is a cell, it contains nucleus in the center, right? Nucleus. Inside the nucleus, let us say this is a human being cell, just skin cell you take and see. Inside that you have a big, let us say a nucleus in the center. Inside the nucleus you have thread-like structures. Thread-like structures, they are called chromatin, right? So how many pairs do we have? 23 pairs, correct? 46 chromosomes. So this is first pair. First chromosome, because we have one pair each. It is just like your shoes or your chappals. You have one pair always, right? One set. So both are, one is left, one is right, but both look alike, right? Does anybody have shoes, a pair of shoes which look different? One looks one color, another looks one color. Maybe that fashion will come in future, but as of now we don't have that fashion, I believe. Right now we are wearing same kind of shoes. So they are only different because one is left, one is right, but both are same in size, same in color, same looking, right? So same way, this is chromosome number 1, that is also chromosome number 1, but one has come from father, one has come from mother, hence they are homologous, right? Both are being called as chromosome 1. Why? Because basically chromosome, if the small chromosome I enlarge it like this, chromosome is a vehicle of heredity, we say. Why? Vehicle. Why vehicle it is called? First of all, we'll understand. Vehicle means a bike or a car. Okay. So let us say you have a bike. You know, you're sitting on the bike and your friend is also sitting behind you. You both are going. So this vehicle is carrying you two people, right? I can give an example where on your bike you are sitting, your friend is sitting, another friend is sitting. But you know, triple riding is an offense. It's not right to do that. So I'm not taking the example where 3 people are sitting on the bike, only 2 people, because you must follow. Generally, teenagers generally do this. They go 3 people on a bike, which is not correct, right? So let me take this small example where only 2 people are going. So basically, vehicle is something that is carrying people, right? So just like a train or bus, you see, bus takes many people on it. Then why is chromosome called a vehicle? Because it is taking who? Genes. What are sitting on the genes are there. So at every location, in fact not on the chromosome genes are there, chromosome has DNA inside, double stranded DNA, right? So double stranded DNA. So at every point of the DNA, small bit of DNA is nothing but gene, isn't it? Small bit of DNA is gene. Fine. Therefore, maybe this particular is gene number 1, not 1 again, because chromosomes we are giving 1 to 3. Here let us give some other name. ABC, let us say. A, B, C, D, like this. So this is one location on a chromosome. This is, let us say, chromosome number 1. On chromosome number 1, at this particular location, I have gene called A. Okay, I have gene called A. On this one, on this chromosome also, in that location you will have the same gene. Let us say this gene is responsible for eye color, let us say so. That means this gene will decide what color your eye should have. Okay, so where is it? example, this is not the correct example. Don't think that your eye color gene is present on chromosome number 1. I am not saying that. I am giving you an imaginary example. Let us assume that eye color gene is present, let us say, on chromosome number 1 at this location. Now, if I see its homologous chromosome, at the same location I will have a gene. That gene will also code for eye color only. So at same location, same genes. Means they will code for same character. Character means eye color, height, skin color, they are characters. That character, how it is different. What I mean is, on this chromosome, capital A may be there. On that chromosome, small a may be there at the same location. That means its dominant form may be here. On homologous chromosome, its recessive form may be there, right? So both still capital A or small a, either it is capital A or small a, both are coding for one character, that is eye color. It may be brown color or it may be blue color, color different, but eye color only. So they are the same gene but different forms of a gene. That is what is allele. So they are alleles. Alleles are nothing but forms of gene. That's it. Understand? Right. It is just like you have a pair of shoes. One is the left side, one is the right side. So left and right shoes are alleles of your shoes. Your shoes are the gene. Shoe is the gene. Shoes is the gene. For that you have two types, two alleles. Okay. Okay, so that is what. Now, combinations. On both homologous chromosomes you may have capital A. On both you may have small a, or one may have capital A, another may have small a, or that one only may have capital A and this one may be small a. Okay, so this is one chromosome, this is another. One comes from mother, one comes from father. So which brings what, we don't know. And that is what we are going to study, how it happens. Second chromosome, let us say. Third chromosome. Fourth chromosome. Like this up to 23. Okay, we have 23 types of chromosomes. Total 46 chromosomes. Number is 46. Types are 23. Okay, what is genotype? Genotype is, now I told you, you have one gene can be expressed in two different forms, right? Alleles. So, for example, if the gene is capital T, gene for tallness, short gene will be small t. That is allele, another form of that gene. Please try to understand, allele is not something else. Allele is also a gene. Allele is a gene. Now, what is small t? It is a gene. Because if I take a chromosome, in that I see DNA. On the DNA I have one particular gene, piece of DNA. It's a gene. It is causing short stature, but still it's a gene. Another chromosome, that part may cause tall stature, but it is also a gene. But two different types, two traits, or what we call as two forms of the same gene. So allele is basically a gene, but a type. Allele, when you say allele, it means there's something else. I mean, there's another form of it, right? Now, in an individual, because we are diploid, we can have two homologous chromosomes. So, one allele here, another allele there. Sometimes it may be same, capital A. Now, your genotype is about writing what alleles you have on both the chromosomes. You write it together, that becomes genotype. Capital T, small t is the genotype. Okay, because on your one chromosome you have capital T, another small t. If you had like this becomes your genotype. If you had like this becomes your genotype. Well, we are only talking about one character, the height, human height. Only for height if you are talking, this is the genotype only for height. But how many characters we have in our body? Height, skin color, eye color, hair color, so many things. You know how many characters? Infinite. Millions of characters we have in the body. So many characters we have, and we have genes for all of them. If you go on writing all of them, space is not enough, right? But a genotype, therefore we are talking about genotype of a particular character, right? Also it depends upon the cross. If we are doing a monohybrid cross, that means we are studying only one character. If we are using a dihybrid cross, you are studying two characters at the same time. For example, in a monohybrid cross, generally you will be studying only one character, height. Okay. In a dihybrid cross, you may be studying some other character also, maybe shape. Like this. That means when I am studying two characters at the same time, one is about the height, another is about the shape of the seed, whatever. Height of the plant, shape of the seed, anything. Two characters. These are two parents. Two parents. This is one parent. In one parent, this is the genotype of that parent. So how did I write this genotype? Look at this once. It means for this parent, if you look at his chromosomes, homologous chromosomes, what you have is capital T is there, capital R is there. For the height gene, he has capital T on one chromosome. He has capital T also in another chromosome. He has capital R on one chromosome, capital R on another chromosome. That is why he became homozygous dominant. That will be the case in another parent also. Small t, small t, small r, small r. Okay, you understand, I'm just trying to relate everything. So some students don't have basics at all in genetics, and this session is especially for them. Someone who might have good knowledge, they might feel that these are basics, but always remember, whatever highest knowledge you have, still it will be dependent on basics only. If you have to build 100-story building, what has to be strong? Foundation, right? You know what? Suddenly, let us say today you have built a house of 5 floors with some foundation, some basement, and tomorrow you got lots of money. You want to build 20 floors above these 5 floors. Do you think you can build it? No, because the basement which you had put was enough only to support 5 floors. It cannot support another 20 floors. So how much you want to build depends upon how strong the basement is. So basic concepts are very important. If they are very strong, you can understand even complex things. So please pay attention to this. So what Mendel did first was he did a monohybrid cross. So he proposed certain laws, law of dominance and law of segregation, law, that is purity of gametes, and then law of independent assortment, which he did later. So what he actually did was, first of all, there are certain things that he did right, okay, but unfortunately after Mendel's death, people understood his importance. As long as he was alive, nobody knew what he did was of phenomenal importance, right? So he actually did some experiments on the pisum or the pea plant, and he selected some 7 characters. And luckily enough, the 7 characters were independently assorted. What does it mean? Let me not talk about independent assortment at this level, because if I talk about it, you'll not understand. First, let me explain it and then talk about it. So let us take a typical monohybrid cross and see. Let us not look at these numbers or the value, whatever I'm writing here. Let us imagine real-time scenario. Let us imagine a tall plant in front of you and a short plant in front of you. You cross both of them. The resultant will be, the result was, see, please remember this. Imagine the output, because when you take a tall plant, does it come written with capital T on it? No, right? It's what we are representing it. So when Mendel took a tall plant, he knew it was a tall plant. When actually he did selfing on them, self-fertilization, so that they get dominant. Anyway, that's a different thing. Pure line selection. So pure lines he created first. So capital T, small t. So they were homozygous. When you cross them, you still got a tall plant. So tall, short. The result is tall. Let us say today you did not know any genetics. You took a tall plant and a short plant and crossed them, you got a tall plant. What will you think? your understanding is like, oh, tall is the dominant. Tall feature is the dominant feature. It suppresses the short feature. Or you will actually think shortness is eliminated. It's not even there. This phenomenon people knew for a very long time. They knew that if one animal had superior qualities and another animal did not have superior qualities, still if you mate them, the offspring has a possibility of getting superior qualities. Okay, so they already knew this and they were doing this artificial breeding for a very long time. So what Mendel did was do a little experimentation on that already known fact. So when he did that, he got only tall plants. So naturally anybody will think that okay, maybe the short trait is totally eliminated now. Now whoever this tall plant that we got, when it gives birth to another offsprings, that will never have short plants. This is what our understanding will be, because shortness is gone, eliminated, we can't see it here. Then just like that experiment, we'll repeat another experiment. What we did first was tall versus short. Again we are doing tall versus short. Okay, so what would you expect? What would you expect? When you crossed tall and short, you got tall, all were tall. Now when you cross tall and short, all should be tall again, because you're just repeating the experiment. But no, this time he had some different results, right? So the results are what we know now. Basically this has to be done. This is a test cross. I'll not go with this basically. First I'll see what Mendel did. Then I'll come to this, so that this is something I wanted you to understand. So what Mendel did was actually with the same plant of this F1 generation, he crossed it. In that cases, what will you get? This was also tall. So what should you get? This is another experiment. This experiment is more valuable because here in fact you are crossing both tall. You should completely get tall plants only. There is not a doubt that you would get a short plant at all, right? But what is the result? Capital T, capital T, capital T, small t, then capital T, small t, small t, small t. 1/4 of all plants was short. Okay, first of all, when Mendel did this cross and he found that all were tall, short is eliminated. Then he said, oh, some characters are dominant over other characters. Thereby he proposed the law of dominance. Now, at F1 level, when he did the F2, when he got this result, then he understood, oh my god, short trait, which I thought disappeared, did not actually disappear. It was there. It was properly preserved somewhere. And when it is given another chance, it is manifested here. Okay. That means now we understood that when these parents produce gametes before sexual reproduction, before fertilization, in those gametes, these traits are properly getting segregated and they're preserved. They're not getting mixed up. Okay. So that segregation of gametes or law of purity of gametes, he proposed when he saw the F2 generation. Please, these are the questions. Now, you might be thinking that I'm not asking questions, but the way I'm teaching is, every point is a question. All you have to do is understand it. If I have to ask questions, you know how they will be? Mendel proposed the law of segregation or the purity of gametes by looking at what? This is a question. Can you say? Options will be by looking at the tall plants in the F1 generation, by looking at the tall plants in the F2 generation, by looking at the short plants in the F1 generation, by looking at the short plants in the F2 generation. Now tell me the answer. So what is the answer? Very good. F2 generation short plants. So when he observed that short plants were again appeared in the F2 generation, then he realized that, oh, the gametes are pure. They never get mixed up. The traits are pure in the gametes. Very good. That is how you have to answer. So law of dominance, when did he propose? When he saw the tall plants of F1 generation. Now, when did he propose independent assortment? And what is this independent assortment all about? Let us see that now. When I am teaching about independent assortment, parallelly you will also learn about linkage. I know linkage is a problem for many people. Now it is going to be very simple. Remember meiosis, the meiosis or meiosis, whatever you pronounce it as. So some pronounce it as meiosis, some as meiosis. So this meiosis or meiosis is a reduction division. What happens here? In the cell, let us take a cell with less number of chromosomes, just to simplify it. So I am taking a case where just 3 types of chromosomes are there. Okay, first chromosome 2 copies, second chromosome 2 copies, third chromosome 2 copies. Or if you want, let me take another 4. So it's diploid. 2n is equal to what? 2n is equal to how many chromosomes? 8. So let us say there is an organism where 2n is equal to 8. Okay, its diploid number is 8, haploid number is 4. What is the genome? Genome. Tell me someone, for example, for this cell, what is the genome? How many chromosomes is genome? Genome means haploid set of chromosomes. N is nothing but genome. Genome is haploid set of chromosomes. That means in this case 4, right? Yeah, some people have answered it right. So that is what a genome is. Fine. Now, this case, let us continue. Let us say this is its DNA. Okay. Now this is the DNA in this chromosome. Okay. Let me take these 2 chromosomes separately and see how crossing over occurs in meiosis 1. Okay. So let us say this is chromosome 1, okay, and now it is undergoing what? It is undergoing crossing over. So these 2 non-sister chromatids, let us say they exchange their genetic materials. After exchanging, how will it look? So can you just make out this difference? Is it visible? So what I've done is, they both have exchanged genetic material. Okay. So now you see, compared to the original, compared to the parent cell that was undergoing meiosis, before undergoing meiosis, this particular chromosome DNA had entirely green DNA, this had blue DNA, let us say. Now in these two chromatids, this is one chromatid of this chromosome, this is another chromatid of this chromosome. Between them, exchange of genetic material occurred during crossing over in the meiosis 1, right? So during that phase, what's happening is, a part of this DNA or chromosome is getting attached here, and a part of chromosome is going there. Because of that, now this chromosome is entirely changed in its structure. You can see, first, this is a pure arm. In this, still entire genome is green. Just colors I am giving you. This entire DNA is pure, but this DNA is recombinant, because here at one point it has a mixed DNA from the other side, from the homologous chromosome it got it. For example, what happens is, if let us say there was a gene called A here originally, okay, so here also A, right? Let us say there is a gene called B here, okay, originally there used to be B here, let us say, and here homologous chromosome, so let us say instead of A, capital A, here it has an allele small a, here also small a, because these two chromatids are nothing but in S phase they got replicated, same DNA. This is a chromosome with the two chromatids, so it will have same DNA. Here small b, let us say, here small b. But now, because these two arms got exchanged exactly at this level, they got exchanged, because of that this capital B will now go there, so this will become capital B, and this will become small b. This will become small b. Understood? So therefore, I can say this entire chromosome, because of taking part of that chromosome into this is a recombinant chromosome. That is also recombinant chromosome. But if you closely observe, within this chromosome also, this chromatid is not recombinant. Only this chromatid is recombinant. Okay. Please remember, very important. Now, in meiosis, what happens is, during the anaphase, this entire chromosome will go to one daughter. This entire chromosome will go to another daughter. Let us draw that in the picture and see how it happens. So let us say this part I got it from there, and another, this part is also, that got it from here. So they exchanged. Now this is chromosome 1 and 1. Okay, like that you have second chromosome also. Here also recombination occurred. Here also recombination occurred. And even here you have recombination. So this is third chromosome and this is fourth chromosome. So crossing over occurred in all chromosomes between the homologous. They exchanged genetic material. Now during anaphase of meiosis 1, what happens? Exactly, chromosomes will be distributed. No centromere splitting, right? In meiosis 1, centromere does not split. Directly chromosomes, whatever are there, they get distributed into half-half. So this entire thing has 8 chromosomes, but the resultant gametes will not have 8 chromosomes. This gamete will get, let us say, this chromosome goes here, let us say. Let me name this was the original parent, no? In that parent, for the first chromosome, it has paternal P, maternal M. First chromosome only paternal side, maternal side. Let us say this is paternal, this is maternal for second chromosome. Same paternal, maternal. Pay attention, it is very important here. Again paternal, maternal. Now, during distribution of chromosomes, first thing is every gamete should get one type of all chromosomes. Otherwise the gamete will not be useful. That gamete will not be viable. What I mean to say is, see how many total chromosomes? 1, 2, 3, 4, 5, 6, 7, 8. If you divide both equally, both will get 4, 4. But how they should be divided? First chromosome should go there, second should go, third should go, fourth should go into one gamete. And again here also you should have all 4 types, 1, 2, 3, 4. It is not like these two of will go there, first chromosome both will go there, and then second will go, and third will go, and nothing from the fourth will go. No, that is not how it happens. Those gametes will not be viable, because for this particular parent, to the individual to survive, it needs 4 types of chromosomes. So each copy from all chromosomes should go. If it does not go, it will result in aneuploidy, that we'll see as a chromosomal abnormality later. So all one copy should go. Now my question starts to you, observe carefully. For the first chromosome, paternal chromosome went into this gamete. We can have 2 gametes out of this, right? Each will carry 4 chromosomes. We are talking about meiosis 1, at the end of meiosis 1. Question is, paternal chromosome entered here from chromosome number 1. Now from chromosome number 2 also you should get 1 chromosome. Which one will come is the question. This is chromosome number 2. Paternal will come or maternal will come? Tell me the answer. From first chromosome, which entered that gamete? Paternal entered. From second chromosome, now which will enter? Paternal or maternal? So tell me the answer quickly. So Garima Sharma says maternal. Any other answer? Any other answer can I see? Paternal, someone is saying. Uma Maheshwari says paternal. So, okay, most answers are paternal. So, shall I tell you the answer? Sai says any one of them. And Meena also says paternal. Okay, let me answer now. Out of all these, the correct answer is that of Sai. He said any one of them. That is what we need to understand. See, first chromosome, paternal copy entered into gamete. From second chromosome, which copy should go? It does not depend on the first chromosome. Why? Anything can go. This is like playing the toss game. Okay. For example, here you have like paternal is heads, maternal is tails. If we toss the coin, which will come, heads or tails? 50-50 chances. Anything can come. So for the first chromosome, the toss worked like the paternal went. For the second chromosome, if you toss, because first time you got paternal, this time you should get maternal or what? Is there some logic like that? No. This time also it depends again. It may be paternal or maternal, anything. Right. So, in the first homologous chromosomes, which chromosome goes into one gamete, that does not influence which chromosome will come from the next set of chromosomes. Understood? Therefore, every pair of chromosomes, how they go into each gamete, are independent of the other pairs of chromosomes above or below. This is independent assortment. Understood? This is independent assortment. So, how it does not mean that in the second pair of chromosomes also paternal should go or maternal should go. Anything can go. This is independent assortment. And this also produces recombinations. Why recombinations? Of course, because if this gamete carries first chromosomes, this one, and that gamete can carry other paternal or maternal, that means the gamete that I'm going to get here, what kind of gamete I'll get? I don't know, multiple combinations can be there. Now imagine how many combinations of gametes can be formed. Maybe from the first chromosome you can have paternal, right? And all other chromosomes may be maternal. Or first may be paternal, second may be paternal, other two may be maternal. Or first three paternal, last one maternal. Or first maternal, so many combinations you can imagine. So you can create, there is mathematics for that also, but right now we are not concerned about that. But we just need to understand that a gamete can be of variety of types, okay, just because of independent assortment. Therefore tell me, independent assortment is giving rise to variations or not? Independent assortment is giving rise to variations or recombinations in some way, right? But first recombination, where did it start? It started with crossing over, right? If crossing over did not happen, this piece wouldn't have gone there, that piece wouldn't have come here. Because of these pieces being exchanged, now the chromosomes got changed. They are not looking like how they were in the original parent. They are already changed, they got exchanged among themselves. So first stage of recombinations happens due to crossing over. Second stage where recombinations happens, independent assortment. Okay, so that means the gamete that you're going to get, how different it will be from the original parent. That difference first starts in crossing over, due to crossing over. Next you will get more difference due to independent assortment. There is another level, third level, where the child that is going to be born to the parent will be entirely different because of another factor. Which is it? Now my question is actually going towards evolution. In evolution chapter you learn this, that variations are produced in sexual reproduction because of three things. I have covered two things now. First is crossing over, like how the parent or father or mother child looks different from them, not exactly same, right? Different. These differences are due to three steps, three reasons. This is a excellent question and excellent clarity of concept that you need to get if you're reading genetics. If I am concluding my session for today with this topic also, I don't feel bad, because if you understand this logic, you can understand every damn thing in genetics. Nothing is there that you cannot understand, because this is the crux of everything. So tell me, first step for variation formation is in crossing over. Second step, independent assortment. Tell me the third step. Anybody? How a child is different from parent? Because of three things. One is what? Crossing over. Another, independent assortment. What is the third thing? Let me see if somebody answers. Someone said mutations. Two people said mutations. See, mutations is totally another way. I did not, my question you didn't listen properly. A child is different from parent because of sexual reproduction. In sexual reproduction, what are the three stages I am asking? Mutations is generally happening. Even in asexual reproduction also, children may be different from parents. That's because of mutations, even in asexual reproduction. But in sexual reproduction, why? So basically, variations are caused due to two reasons. First reason is mutation, second reason is this one, what? Sexual reproduction. So keep mutations aside. I'm talking about in sexual reproduction also, what are the stages, why sexual reproduction is causing variations is my question. So three stages. One is crossing over, another is independent assortment. What is the third one? The third one is fertilization. Write this point down. Important. Fertilization. Fertilization. Because during fertilization, which gamete, and see, like you have seen, since there are so many types of chromosomes here, which chromosome will enter which gamete you don't know. So you can get millions of types of gametes. A lot of types of gametes you can get. And if as in a single parent you are getting this many types of gametes, we are still talking about a single parent. Let us say this entirely male parent. Similarly, female parent is also forming gametes. That female parent will also form variety of gametes. Male parent will form variety of gametes. Ultimately, which two gametes come and fuse, that becomes zygote, that becomes the child. So fertilization is the another one that will decide how the child is going to be different. So that is variations. So remember this. Okay, crossing over, independent assortment and fertilization. These are the three things which decide how the child is going to be different from the parent in sexual reproduction. Somebody answered recombination. Please try to understand, recombination is a term that is generally used for crossing over. Generally used for crossing over. Okay. If you're talking about variations for that we have two reasons: mutations, sexual reproduction. Under sexual reproduction, variations are caused by crossing over. That's nothing but recombination, crossing over, and independent assortment and fertilization. It's clear, right? Let us move further. Now, if paternal comes here from first chromosome, anything can come from second, anything can come from third, anything can come from fourth. Fine. Now you see, this paternal chromosome I have drawn, but I have to draw this part also, right? It has undergone recombination here. Like that, this has undergone recombination here. Now, to this gamete, this is just a product of meiosis 1. It has to go through meiosis 2. What happens when it goes through meiosis 2? Let us see. Sorry, this is not the picture that I have to draw. So that gamete already had only 4 chromosomes, right? And those chromosomes, each of them were already recombinants. Now this gamete is going through meiosis 2 to produce the final gametes that we have. Meiosis 2. This was first chromosome, second, third, fourth. You all know that meiosis 2 is just like mitosis, right? That means splitting of centromere will happen, right? Very interesting observation I want you to observe here. I want to show to you. Probably nobody has told you in the classrooms, maybe very rarely someone might have told you, but generally I observed no teacher actually tells you this. I can frame a question on this actually. The question is, meiosis 2 and mitosis, or the question is this: meiosis 2 is similar to mitosis. Okay, this is assertion. What is assertion? Meiosis 2 is similar to mitosis. Okay, is it correct? The process of meiosis 2 is similar to that of mitosis. Is it correct or not? First of all, it's a correct statement. Okay, reason: the cells that are formed from meiosis 2 are genetically identical to the immediate parent cells that went through the meiosis 2. Is it correct? This is the entire meiosis, isn't it? This is meiosis 1. This is meiosis 2. Okay. So here this is the parent cell. Compared to this parent cell, these two daughter cells are the same? No. Because they have 8 chromosomes. These two have only 4. So genetically they are not identical. Meiosis 1. Meiosis 1 is totally not, it is not at all like mitosis also, because there is no splitting of centromere. But meiosis 2 is just like mitosis, we know, because here these 4 chromosomes, they split into, each chromosome splits into half. Therefore we will again get 4 chromosomes each. Ultimately 4 and 4. Fine. So can I say that these two cells resemble its immediate parent? That means this, not this one. This one. Immediate parent is this one. Can I say these two daughter cells resemble this one? This parent cell which went through the meiosis 2? Can I say this or not? Answer this question. Because they have 4, this also have 4 chromosomes. Are they identical chromosomes? This has, are they the same chromosomes? That is something you have to question. Same chromosome only got into 2 pieces and went into 2 daughter cells. So maybe it will be identical? No, it won't be. See why. Here you see, we are actually writing that process. This cell is this one. This cell, we are drawing it there. It has 4 chromosomes. Yes, 4 chromosomes. But it has 2 types of chromatids. See, one is a pure chromatid, one is a recombinant chromatid. One is a pure chromatid, one is a recombinant. Here also pure recombinant, pure recombinant. Now, when the centromere splits into 2, I split like this into 2. Now you see what is going to happen. This chromosome, this chromatid, pure chromatid will go here, let us say, for chromosome 1. This recombinant will go here. Fine. For the second chromosome, let us say, the recombinant is going here. Anything can go again. Then the pure should go here. For the third, let us say the pure is going here. Okay. And the recombinant is going here. For the fourth one, let us say the recombinant is going here. And the pure is going here. Now you see, compared to the original, are they similar? They are not. When they split into, again when they enter the G1 S phase and they split into chromatids, this chromosome, when it splits into chromatids, you will get 2 pure chromatids. This will get 2 recombinant chromatids. Recombinant chromatids. Pure chromatids. So just observe any one chromosome. First chromosome, pure chromatids. Its parent had 1 pure, 1 recombinant chromatid. So the daughter is not same as the parent. Then why are we still calling it mitosis? Because the process is mitosis, where splitting of centromere occurs, each chromosome 2 copies will go to daughter cells. Only that much. Only the process looks like mitosis, but the genetics is different again. In mitosis actually, the recombination crossing over doesn't occur, right? No crossing over occurs. All will be pure chromatids only. So which chromatid goes where, still they look like parent cell. So this observation is a fantastic observation. I want you to understand this. Someone asked, is there any crossing over in the sister chromatids? No. Only among the non-sister chromatids there will be crossing over. And also, the chromosomes generally we draw like this, no? We draw side by side like this. Okay, like this. And we will show that these 2 chromatids are crossing over. They don't stand side by side. They are not performing a theater in front of you on the stage. So it's not like let me stand here and we will cross over. No, they stand one upon the other like this. Actually, they attach like this. But how can I draw that on the board? It's very difficult, no? They attach like this. If I draw one, behind it there will be another. How will you see that which is behind it? That is why we are drawing side by side. Otherwise, this will actually come and fall on this. It will attach to this. In between them, there will be a synaptonemal complex, protein complex that will help their attachment. And remember, this chiasmata formation, for our convenience, we are drawing only chiasmata at this level. But chiasmata can form at multiple levels. At multiple levels, crossing over may occur. And not just 2 chromatid arms will take part. Sometimes these 2 also may take part. In this diagram, you won't believe. You will think these 2 are close, no? They are crossing over. These 2 are very far away, no? How will they cross over? This doubt you will get. But this is not the right accurate diagram. This is what is given in your textbooks, but the original diagram will be, if these are the bottom parts you see, these are, then another one will sit on that. Because of that, all along the length anywhere crossing over can occur, and between these two chromatids also crossing over can occur, but mostly only between two chromatids crossing over occurs mostly. This is the thing. Okay, so due to the lack of time I have to stop the live class here, but in the next class tomorrow I will continue this. I'm going to see you all, I hope, for the next class, because I'm sure you don't want to miss these wonderful concepts. I'm enjoying this class and I hope you're also doing that, because tomorrow I'm going to teach you linkage. You have to come at least for linkage, right? When you understood independent assortment, now you need to understand what linkage is, because you have studied so much. Why do you want to miss out on that path? You have already gone to the deeper part. Only a little bit is remaining. Let us even learn that. Not only that, somebody is asked for sex determination. I will also cover that. Don't worry. Tomorrow, like first half an hour, I'll try to cover all these concepts, and then we'll move on with the topic of tomorrow. Okay, so we're little learning, I will letting going off the schedule. We're just a little delaying stuff, but thing is I don't want to miss anything. I can say that okay, this is what I can do in genetics, I'll move on to another topic. Somehow I can't do that, because I can see many students are following me regularly, so I feel like you are my students now, so I have to teach you genuinely whatever I know. So I hope you're studying well, and you have any doubts, please go to Toppr app, and you have doubts on chat there. Ask any doubts there. Our teachers are always ready 24/7. They'll answer you instantly, within seconds, within minutes you'll get the answer, and the right answer you'll get it. And also practice as many questions as possible on Toppr app. And you know something, all the videos on Toppr are free, completely, all the videos. So any topic, anything, you can just watch it for free. Don't miss this golden opportunity. I want you to crack NEET exam and come out with flying colors. So see you in the next live class. Until then, signing off.